Good afternoon, everyone. Welcome to our CGC webinar series. So today we're showcasing the work of two more of our poster winners from the CGC annual meeting last year in August. We have Dr. Nicholas Semenkovich and Sharon Freshour, both of who are from Washington University in St. Louis. But before we start, the CGC would like to remind everyone that our annual meeting this year will be from August 13th to the 16th in St. Louis. So if you have any original genomic findings from a clinical or research setting that you would like to share, we encourage you to submit an abstract. We would also like to invite trainees and technologists to apply for the Trainee and Technologist Awards. Abstract submission is open, and the deadline to submit is next Tuesday, February the 28th. And as always, we would like to encourage those who are interested to join the CGC to do so. There are a number of benefits, such as being able to connect with new colleagues, initiate collaborations, and also access educational opportunities. It also allows you to serve on a committee or a working group, gives you reduced registration for the annual meetings, and you also have free online subscription to the Journal of Cancer Genetics, among other things. So please check out our website for more information regarding this. So with that, I would like to get started and introduce our two speakers for today. So our first speaker is Dr. Semenkovich, who is a physician scientist and clinical fellow in endocrinology, metabolism, and lipid research at the Washington University in St. Louis. He's currently a postdoctoral scholar in the lab of Dr. Adel Chowdhury, working on cell-free DNA with a focus on developing machine learning models to analyze it, and also to provide insights in chronic and metabolic disease, in addition to oncology. Thank you, Nicholas. The floor is yours. Just stop so you can share.
Thank you all for that wonderful introduction. Let me pull up my slides here. I trust you all can see them. So thanks to the CGC for a poster award at last year's conference. And I'm here to present today some still in progress work on ovarian cancer ecotypes, which is what my poster was on last year. Thanks again to the CGC for inviting me to talk here. I'm going to give you all about 20 or so slides, talk for at most 20 or so minutes about a project that I've been working on with Adel Chowdhury, my PI, who's in radiation oncology, and Catherine Fu, who I think recently moved to UCSF and is in GynOnc. We'll be talking about ovarian cancer ecotypes. This project spurned out of the concept that ovarian cancer is very highly lethal, and there aren't a lot of good targeted treatments for it. Ovarian cancer is the 5th leading cause of death in women in the United States. You can see on the right panel here, it is right in the middle of the pack, about 4% of cancer mortality associated with ovarian cancer. And I'm working in the lab of Adel Chowdhury, and there's Catherine Fu as well on the right, who we owe a lot of these samples and guidance in this research to. Ovarian cancer survival in particular is much worse than other cancers that I think are often highlighted in the media. So here is some data from the often cited 2022 Cancer Statistics Summary of Cancer Mortality. On the left and right, there's both female breast cancer and there's prostate cancer on the right. And the thing to note from these slides is that in general, at all stages of diagnosis, although many cases of breast cancer and certainly prostate cancer as well can have very high mortality, across all stages, the average survival is generally pretty good. The 5-year relative survival in breast cancer is essentially 90% across all stages at presentation. The same is true in prostate cancer. Things, however, are much worse in ovarian cancer. You can see the average survival across 5 years is less than 50% across all stages. Probably the big reason for this is that most patients present with what may be widely metastatic disease. So unlike both breast and prostate cancer, there are no good screening technologies or screening approaches that are applied at a population level. And so many women who come in with ovarian cancer already have widely metastatic disease, in which case the therapeutic options are limited. So this is a study in particular of one of the most common types of ovarian cancer, specifically high-grade serous ovarian cancer, which you might see abbreviated as HGSC or HGSOC within this talk and within the literature. And my goal here was to better understand the tumor microenvironment and the cells that were part of tumor ecosystems interacting mostly with the immune system. I did this project in part with the collaborator, Dr. Dave Much at the top, who's one of our GynOnc surgeons here at WashU. And we had unique access to a consortium of samples that were both pre- and post-therapy samples. So when women are diagnosed with ovarian cancer, uniquely at the start of diagnosis, they usually have a laparoscopic surgery where they undergo a biopsy, frequently of a mental metastatic disease. That's what you can see in the picture here on the picture here on the left. It's an individual undergoing a biopsy of one of these pre-therapy samples. Then they undergo usually a neoadjuvant chemotherapy followed by a cytoreductive surgery. so a combination of both surgery and chemotherapy. And then these patients frequently undergo a subsequent surgery or subsequent biopsy to assess their response at times. What's interesting about high-grade serous ovarian cancer is within the literature and a lot of physicians treating these patients note that there tend to be groups that are both responders and non-responders. A number of patients seem to respond very well to initial chemotherapy. A number seem to have almost no response to chemotherapy. And on the top, you see a patient who has what appear to be very few visible omental mets. But on the bottom right panel, you can see a patient who's a non-responder to their chemotherapy and cytoreductive surgery, a lot of metastatic implants within the omentum. So this project focused on high-grade serous ovarian cancer with both single and bulk RNA-seq data from patients with high-grade serous ovarian cancer. We had access to patients who are undergoing chemotherapy, frequently carbotaxol, followed by a debulking surgery. We refer to some of these patients as NEO, sort of neoadjuvant pre-chemotherapy and surgery samples. And then some ICS patients, those who have undergone interval cytoreductive surgery, both post-chemotherapy and after their surgery. And we have bulk RNA-seq samples from about 46 samples as well, so from 23 patients who have also undergone the same. So pre-chemo and surgery, and then sort of post-chemo and surgery, as well as a subset of single-cell RNA-seq samples. We had 11 at sort of the start of the project. This is expanded a little bit more now, but I'll be talking about the initial beginnings of the project. And a lot of this work was done by now Dr. Emily Kotnick, a PhD scientist who recently graduated from Dr. Fu's lab, and by Abul Usmani, one of the staff scientists in Adil Chowdhury's lab, as well as an additional staff scientist and Andrew Shannon undergraduate who's worked closely with me on this project. At a high level, my analysis project approaching this was to ultimately try to implement something called ecotyper to identify co-occurring transcriptional states, to try to find out when different transcriptional patterns were seen across multiple cell types within these data. How I did that was I took our input single cell data, as well as some external samples of single cells, some additional collaborators with single cell samples, and fundamentally focused on trying to define the ground truth cell states, which is the most challenging part of this project. And I'll talk very kind of work in progress today of how that took place. The idea is we have a lot of single cell data. The question is what individual single cell is assigned to what ground type cell? Is this cell a lymphocyte? Is this cell a monocyte or a macrophage? Is this cell a tumor cell? And how can we define that? And building that fingerprint of cell states was critical for trying to figure out how cell types are interacting. I also used a variety of other tools that I'll talk about in the next few slides, a series of markers and predictors of copy number variation to try to identify which specific cells were tumor cells versus normal host cells, cytotrace to predict developmental maturity of these cell types, and then also helped train this model using some bulk RNA-seq data, both from our internal samples and some TCGA samples as well. The goal of this project ultimately was to apply a framework called ecotyper that I think probably a lot of you have seen or have used. This is from a collaborator that we have from Adil Chowdhury's group out at Stanford, Aaron Newman. His group built a framework that tries to identify cell states across cellular communities that are shared using both bulk and single cell data. And the idea here is that if you have a collection of different cell types with lots of different transcriptional patterns, maybe you can see if those patterns all go up or down or somehow associated by different parameters. Maybe they might be interacting with each other. You could predict that, hey, if you have these tumor cells, and whenever these tumor cells are expressing these unique gene signatures, and at the same time, maybe the macrophage population that you see is expressing these gene signatures in samples, you could say perhaps these are associated with each other. Perhaps these make up an individual tumor ecotype. And then ultimately you have to prove that later on, either through spatial transcriptomics or through some other approaches that we're taking. These models were originally trained on bulk carcinoma data and on lymphoma data. If you've read the original ecotyper paper, I think it's kind of worth taking a look at. And so we've expanded this a little bit in our studies, and the initial one was also based a little bit on ovarian, and that's what we're focused on here today. So to give you a sense of how this worked, I think a lot of you have done single cell data before, but I always like to kind of walk through my analytical approach. We started out in the lab initially using Serous and ultimately transitioned over to using Python to calling ground truths, with the biggest challenge in this project trying to say, hey, we have a lot of single cell ovarian data pre and post chemo. How can we map back individual transcriptional profiles to what the ground truth cell type is? I ultimately applied this using the SCVI tools within Python and used data from some labeled transfer projects from the Chan-Zuckerberg initiative, something called the Tabula Sapiens project that has a lot of expert annotated single cells, to try to figure out what our ground truths were for ecotyper. From our initial samples, just from 11 single cell samples, now that we've expanded a bit, we filtered those to mitochondrial reads less than 20%, a reasonable range of features that were called, part to make sure we don't have doublets and other problems with our samples, and identifying genes counted in at least 3 cells. Afterwards, we had about 42,000 single cells, and then underwent sort of standard approaches through single cell analysis. There's log normalization, filtering for variable features, doing PCA. I don't want to go through all the details of that, but I'll just highlight some of the clusters that we found. And I like to remind people when we look at PCA plots, there's a wonderful summary, if you haven't read it, from Google's PAIR initiative. So the PAIR initiative, which I know has been in the news at times recently, has a good summary on understanding UMAP plots. And I think this is worth a read to anybody going into the field or looking at these plots. When you're looking at t-SNE or UMAP plots, it's important to remember that a lot of the things that strike you visually are totally meaningless in terms of the underlying statistics of UMAP. So things to remember here is that the relative size of an individual cluster is meaningless within a UMAP plot, and that the cluster differences, the actual distances between the two clusters, may also mean nothing, depending upon how you've implemented your clustering and what data is sort of underlying what you're looking at. I think it's always important to remember, because sometimes I see these plots and I'm like, wow, these clusters are so far away, these cell types are very different, and that may not be the case. Ultimately, I myself in the lab and with collaborators used a variety of different approaches to call ground truth across these single cell data. We applied single R, as well as a few machine learning based models to try to identify which cell types were the true ground truth cell types from the single cell data. Single R, I think, is probably the most useful tool, and I'll take a point of highlighting that here. This is an example output from single R. If you've applied this package, you can take an input single cell cluster from your UMAP or t-SNE data, and then get a summary of what it thinks the most likely cell type of origin is. Sometimes it gets a little overly specific. So in this case, I think we had some smooth muscle cells, and it identifies one of the highest ranked cluster here is a lot of smooth muscles, but identifies a lot of things that are sort of closely related, but probably not. We probably think this cluster was not an osteoblast or a foreskin fibroblast. It's probably a smooth muscle cell. And we went on to manually validate all of our cluster labeling. And so using a few databases, including PangalloDB and others, with the help of excellent work from an undergrad, Andrew Chen in our group, went through each individual cell type cluster. This example is one of the malignant cell clusters that we thought were epithelial cells, and overlaid maps for EpCAM, and you saw distinct expression within that cluster, as well as 2 additional markers of high-grade serous ovarian cancer, KRT8 and WFDC2, that were both tightly expressed within that cluster. So we went through and did this manually across all of our cell types, and ultimately built out this project with very reproducible code in Google Colab, so that anyone, when we finally publish this work, will be able to basically click one button and go through and annotate all of our clusters, call copy net number variation, overlay marker genes, see individual annotations, and then basically reproduce all the analysis in the paper with kind of one click, which we're pretty excited about. And then we ended up with a final collection of annotations, calling across all of our single cells and across some expanded data, both CD4s and CD8s, modified macrophage populations, endothelial cells. You can see a map here of overlying marker genes. So that's a lot of very nitty-gritty details, ultimately to validate where our cancer cells were as well. We also applied something called copycat to identify copy number variation. This is a very useful tool where if you aren't sure that your cells are malignant or not, or you want to really validate which ones are malignant or not, you can try to identify copy number variation within your single cell data. We found about 8% of our data were likely malignant cells, and that overlaid beautifully within one specific distinct cluster that was aneuploid while the others were diploid. This was all performed just to do one critical component of the ecotyper framework. And so ecotyper, again, works by taking a large collection of single cell or bulk data and trying to find co-associated cell states and saying that maybe these cell states are interacting with each other in some way. So the important components of ecotyper is that it's essentially kind of a testing and training framework. You kind of test and train your data on your single cell data. You sort of discover cell states and ecotypes. And then once you build a model of your cell states, you can apply that then subsequently to bulk data, something called recovery. So I initially began by using discovery, ecotyper in discovery mode, to train the single cell annotated data sets, and then applied the recovery mode to our bulk RNA-seq data to 23 paired patients. Applying these now in discovery mode, ecotyper identified a number of co-occurring cellular states. We're not quite ready to publish all this work yet, but I'll highlight just a couple of things here, both within monocytes and macrophages. In this particular run of ecotyper, we identified certain co-occurring genes that seem to be distinct within a certain population of monocytes and macrophages. The same was true of NK cells and within our malignant cell types. Once we had built this model in the discovery mode of ecotyper, and then applied this model in recovery mode. So this is taking our built model and applying it to all the bulk RNA-seq data that we have, and we're working on expanding this more now. And then identified distinct ecotypes. In this case, ecotyper calls it E1, E2, E3, basically different transcriptional sets of patterns of expression that were seen across all of our bulk RNA-seq data sets. Now that we have these, we have a number of patients who are labeled as basically assigned to ecotype 1 or ecotype 2 or ecotype 3. And we're working on mapping those back to clinical survival correlates. So we've traced through these patients, summarized their overall clinical response, their survival, any complications that they had throughout their treatment. And our goal is to identify which specific ecotypes and which transcriptional networks are associated with either good or bad responses in high-grade serous ovarian cancer. We're working on now following up a couple of these critical gene hits with spatial transcriptomics. So the challenge with all these and with all machine learning models is that these are predictive, but you don't really know if these are true associations or not. You see that certain genes go up and down at the same time as others or seem to be in co-occurring states, but we need to actually prove that. So we're developing some spatial transcriptomic approaches to kind of overlay what we've seen with these ecotypes and show that there are truly distinct transcriptional patterns that we think are real and have clinical significance. And then we're also working on integrating these with cell-free DNA and other biomarkers in the lab. Dr. Fu's group is also applying these to some patient-derived xenograft models and some tumor cell cultures. This is very much a work in progress, but I think I'm going to stop around here and discuss some questions. Again, thanks to the CGC for inviting me in to give this talk. This work took a lot of a team and is still ongoing. So this was started by Dr. Emily Kotnick, a PhD from Catherine Fu's group, and Abel Usmani, with a staff scientist in Adel's group, along with another staff scientist in our undergrad. And then in addition to my PI, Adel Chowdhury, Catherine Fu, who started this project, and Dave Much, who we owe many of the surgical samples to as well. This wouldn't be possible without a home research team too. It always takes an army. This is my huge army at home. I have baby Candor here, who's like a week and change old now. This is Braden, my 1.5-year-old here, reading about a page kidney. And then Brooklyn and Kendrick here with a chart of what looks like their book money associated with COVID quarantine, when they had COVID in the fall. And the Kendrick here with a little sigmoidal function of his Lego money associated with his COVID antigen positivity. And this is my wife, Tara. She is a cardiothoracic surgeon here, and she is seen here with 1, 2, 3, 4, 5, 6, 7, 8 communications devices. So that is our home army that made this research possible. And with that, I will take any questions anybody has.
Thank you, Nicholas. So I forgot to mention earlier on, if you have any questions, please put them in the Q&A box. I'll just get this started. I have a couple of questions. So the first one is, why are there really no screening options for ovarian cancer? And what does CA-125 play here?
That's a great question. It's probably better suited for somebody in the public health realm. I imagine, from what I know in the clinical setting, CA-125 has a lot of false positives, and I don't think it's particularly sensitive for patients. Certainly, it's used at times when patients present with high-grade ovarian cancer, but I don't think it's used as a screening test, and has a lot of associated false positives. I think it's also elevated in colorectal cancer and a number of other things. Independently of sort of this work, in Adel's group, we're interested in applying cell-free DNA, which has been popularized by a lot of groups in screening for ovarian cancer and other malignancies early on. But that's a great question. I'm sure there's some other groups that have looked into that. I'm not sure the details behind why there's not a good screening test for ovarian cancer.
For the cell-free DNA, are you using peripheral blood, or are you actually looking at ascites, spinning it down and looking at the
we're not looking at that. So we are not looking at acidic fluid. We have broad interest in a lot of diseases for early prediction, and including ovarian cancer, amongst others. We're not looking at acidic fluid because I think the only reason clinically you would have that would be if you already have a diagnosis, or if you're already suspicious of ovarian cancer.
I see some questions now. So first one, is it more of the microenvironment that is facilitating the growth of the tumor?
We would certainly hope so. I think that's what we'd hope to find and show. I think the jury's still out on that. So we're looking into some spatial data to see if we have some good evidence that there's the tumor microenvironment encourages that growth in patients who sort of are non-responders to chemotherapy. It certainly looks like there is some interplay between the localized macrophage population, but it'll take more studies to show if that's the case.
Okay, and continuing from that, the other question is, how much cell-to-cell variability is there within individual tumors, and how many single cells were needed to ensure that you had a representation of a single tumor?
That's a good question. I guess I don't have a good sense of how to quantify. I don't know if there's a good standard in the field for how to quantify variation across tumors. That's a great question. We certainly see at least 2 distinct ecotypes that result from these, which implies that there's not overwhelming variation, at least taken by this approach. And we chose, just practically limited by the number of samples that we had available, which was, as you showed, 11 initial single cell samples that we're trying to expand now. But I don't have a good sense of how to quantify real variation across them.
Okay. Another question is, so in one of your figures, you were showing there was mention of CDH1 and EpCAM.
Mhm.
I'm just wondering, do you see any signatures of EMT process in, say, more aggressive cell
Sure, and
phenotype?
progression to metastatic disease. I haven't looked at that more specifically, in part because a lot of these patients already present with widespread metastatic disease. And so I imagine that you would, but I think we are, by choosing this particular subpopulation, we're sort of filtering for a group that already likely has widespread metastatic disease at presentation. So every one of these patients in the study had, it expanded, peritoneal mets at the time of initial diagnosis.
Okay, all right. Not seeing any questions on that. Okay, well, maybe we'll just move on to the next speaker. And if any questions come up after, we can always ask those.
That sounds great. Thanks so much.
Thank you. Thank you. So we can go on to our next speaker now, Sharon Freshour. She's a PhD candidate in the Human and Statistical Genetics program at Washington University in St. Louis. She's interested in utilizing next generation sequencing and bioinformatics analysis to understand the genomic landscape of cancer and how it relates to treatment response. Her current research topics include using whole genome sequencing to explore copy number alterations in childhood brain tumors, and using single-cell RNA sequencing to understand mechanisms of response to checkpoint inhibitor treatment in a mouse model of bladder cancer. Thank you, Sharon. The floor is yours. And also, if anyone has questions, please put them in the Q&A box. Thank you.
Thanks very much. I'll just go ahead and start sharing my slides. Great. So thank you for that introduction and for giving me the chance to share my research today. So as Shamini said, I'm Sharon. I'm a PhD student at Washington University. And today I'm going to be talking about copy number alterations in childhood brain tumors. So before I get into the details of the specific project I'm going to talk about, I just wanted to briefly mention some background about childhood brain tumors. So they're the most common solid tumor cancer that's seen in children. They're the second most common childhood cancer overall, and they're the leading cause of cancer-related deaths in children. So childhood brain tumors encompass a large range of histological disease types and subtypes. So broadly speaking, they're usually grouped based on the cell type of origin. So for example, cancers that start in glial cells are categorized as gliomas. However, even within those categories, there can be many subtypes of disease. So childhood brain tumors encompass a pretty large range of cancer types. However, one common characteristic that was thought to be shared across these types was that the genomes of these patients are generally considered to be fairly stable. So you wouldn't expect to see much copy number alteration or see very extensive copy number events. So now getting into a bit of the background for the project I'm going to discuss. This project sort of grew out of an unexpected finding from a small cohort of diagnostic samples from here at WashU. So these samples had low-pass whole genome sequencing performed, and then had their genomes analyzed, including having copy number calling done. So these initial copy number calling results showed that copy number alterations were actually occurring pretty often in these samples, much more so than expected. So we decided to sort of explore this finding in more detail in a slightly expanded cohort. So in total, we ended up looking at 66 samples across 4 diagnosis groups, which are listed in this table here. So to look at copy number variation in these samples, of course, the first thing we did was copy number calling. So we did this by running CNV-KIT on those low-pass whole genome samples. And these samples did not have matched normals. So we used a panel of normal samples from a cohort of about 50 breast cancer patients as our normal for the copy number calling. And then after getting those copy number results, we performed manual correction of the CNV calling, which I'll discuss more in the next few slides. And then for all the downstream analysis, we use those corrected copy number results. So these two plots are an example of the plots that we use to do our manual correction. So this is a sample where the genome's totally quiet, so there's no copy number events at all. And the copy number calling did not need any correction. But here I'll just explain the plots a little bit. So the plot on the left is used to visualize allele frequencies across the genome, so we can get an idea of if there's any loss of heterozygosity in the sample and use that to inform our adjustments of the copy number calls. So on the x-axis, we're showing the chromosome numbers, and then on the y-axis, we're showing the allele frequencies. So I know there's kind of a lot going on in this plot, but for the sake of this presentation, what you can pay attention to are these two blue lines in the middle of the plot. And then in the plot on the right, we are looking at the copy number calling results. So again, the x-axis is showing the chromosome numbers, and the y-axis is showing the change in copy number. So this red dotted line at 0.5 is our cutoff for calling a copy gain. So any segments above that line would be called as gains. And then the blue dotted line at negative 0.5 is showing the cutoff for calling a copy loss. So any segments below that line would be called as losses. The percentage shown in the plot title on this right plot is the total percent of the genome that is copy altered. So that's calculated by summing the total number of base pairs that are found in segments of gain or loss and dividing it by the size of the genome. And then the three values that we're going to focus on for adjusting copy number calls are the upper and lower cutoffs, and then what we call a recentering factor. So as I mentioned, this is an example of a sample that had a quiet genome and didn't need correction. So if you look at the blue lines in the allele frequency plot, you see that they generally fall around 0.4 and 0.6, which indicates the sample is heterozygous across the genome. So we would expect that our segments are all going to be neutral around 0 for the copy number calling, which is what we see. So these two plots are showing an example of a sample that had copy loss, but did not need any correction because that loss was correctly called. So if you look at the LOH plot on the left, you can see there's this loss of heterozygosity for what looks like the full length of chromosome 6. And then in the copy number plot on the right, you can see that there's a corresponding copy loss called, again on chromosome 6, and it falls below that standard cutoff of negative 0.5. So this is an example where CNV kit calling was working well on its own, so we didn't need to do any correction. So this is showing an example where the upper cutoff needed to be adjusted, but we didn't need to apply any sort of recentering factor. So in the plot on the right, you can see that there's copy loss on chromosome 6 and copy gain on chromosome 9 that are called using the standard cutoffs. And if you look at the plot on the left, there are corresponding changes in heterozygosity on those chromosomes. However, there appears to be this change in heterozygosity on chromosome 19 that corresponds to a copy gain, but that copy gain falls below the 0.5 cutoff. So it's not actually being called as a gain. So in this case, we decide to lower the cutoff for what we call as a gain. And this plot is just showing the corrected copy number plot for that same sample, with the upper cutoff now changed to 0.3 instead of 0.5, which allows this copy gain on chromosome 19 to be included as a non-neutral call. And then finally, these plots are showing an example where a sample had to have a recentering value applied and had the upper cutoff lowered. So in this case, we decided that a recentering value needed to be applied based on the regions that appear to be heterozygous in the LOH plot, but they're not centered around 0 in the copy number plot. So for example, chromosomes 1, 2, and 3. So to determine the recentering value to apply, we calculated the average copy number value of those regions and then recentered by adding that average value to all of the copy number values across the genome. And then we also lowered the cutoff for calling a segment as a copy gain to make sure to include regions that appear to be true copy number gains, such as those on chromosomes 4, 5, and 7. So again, this is showing the corrected CMV plot for that sample. So as you can see, when we apply that recentering factor, the neutral regions are now centered primarily around 0. And then by lowering the copy gain cutoff to 0.3, we make sure to include the copy gains called on several chromosomes. And I do want to mention here that although the examples I showed only had one of the cutoff values changed, there can be cases where both cutoffs are adjusted and you also apply a recentering factor. So once we had completed that copy number calling and the correction, the next thing we did was look at the extent of copy number alteration across our samples. So here I'm showing the distribution of the percentage of the genome that was altered for each sample. So the violin plot is just showing the overall distribution of those values, and each point corresponds to a single sample with the value on the y-axis showing that percentage of the genome that was altered. So the range of values for this went from 0 to about 50%, with 59 out of 66 patients having some level of copy number alteration. And the average percent of copy alteration was around 14. So again, these results just sort of showed that copy number alterations were pretty common in our cohort, and some cases were fairly extensive. So one of the next things we looked at was whether the percent of the genome that was copy altered was correlated with overall survival across our cohort. So the value at the top of this plot is showing the Spearman correlation value. So just a reminder, the closer a correlation value is to 1 or -1, the stronger the relationship between the two variables, while a value closer to 0 means little or no correlation. So you can see from our correlation value, it's pretty small, indicating there's very little correlation between the percent of the genome that's copy altered and overall survival, at least on a cohort level. So the next thing we looked at was correlation between the percent of copy alteration and survival within each diagnosis group. So you can see that for some of the diagnosis groups like ATRT or high-grade glioma, the absolute value of the correlation increases quite a bit when you compare it to the cohort-wide value. However, these correlation values still don't really indicate a strong relationship between copy alteration and survival. And there also is the caveat, of course, that these groups had pretty low sample numbers, so results could easily be impacted by the presence of outliers. So the next thing that we wanted to do was to try to increase our sample size. So to do that, we decided to use samples made available by the Children's Brain Tumor Network, or CBTN. So CBTN is an international clinical research consortium made up of over 30 institutions. And their goal is generally to advance the field of research in childhood brain tumors. So they've collected samples from thousands of patients across brain cancer types. They've performed multi-omic analysis on these samples, and they've made those data sets available for research use. So in total, we added 265 samples from the CBTN dataset across those same 4 diagnosis groups from the WashU cohort. So these samples already had copy number calling results available from Control3C, which is just another copy number calling tool. So we decided to use those results for our initial analysis. So similar to the WashU cohort, we calculated the percentage of copy alteration for each sample. So this plot is showing the distribution of that copy alteration percentage for the CBTN samples. So again, each point corresponds to a sample. And in this cohort, all samples technically were reported to have some level of copy alteration. Although some of the samples had extremely low percentages that were essentially 0. But we did see similar patterns to those seen in the WashU cohort. So in particular, copy number alteration was very common. And there were some cases that had extensive alteration. So in this cohort, the range of copy number percentage went as high as 94%, with an average value around 15%. So next, we merged the 2 cohorts and repeated our analysis looking at the correlation between copy alteration and overall survival. So similar to the WashU cohort, we see that in this combined cohort, there's really no strong correlation between the extent of copy number alteration and survival. So we then looked at separating the combined cohort by diagnosis group. So again, we still don't see any strong correlation values. However, the absolute value for ependymoma and high-grade glioma increased. So in particular, the value for high-grade glioma went from -2.1 to -3.4, suggesting that the increased sample size could be revealing a true pattern of negative correlation, which would mean that the extent of copy alteration increases in high-grade glioma as overall survival decreases. However, we'd of course need a larger sample size to verify if this correlation is correct. So we next wanted to explore the correlation between survival, copy alteration, and several other variables, including diagnosis group, sex, and agic diagnosis. So to do this, we use the Cox proportional hazards model, which is a regression model that's used to look at the association between survival time and multiple covariates at the same time. So to run this model, we used the Cox pH function in R, which reports a p-value and hazard ratio for each covariate. So the p-value indicates if a covariate appears to be significantly associated with changes in survival, and the value of the hazard ratio indicates the direction of the association. So a hazard ratio greater than 1 indicates association with worse survival, while a hazard ratio less than 1 indicates association with better survival. So in addition to running our model with CNV percent, diagnosis group, age at diagnosis, and sex, we also included cohort as a covariate. So unfortunately, this analysis appeared to show that the only covariate that was significantly associated with changes in survival was which cohort the sample came from. So this indicated that our results were being influenced by batch effects potentially. So this table is showing the p-values and hazard ratios reported for each covariate we used in the model. And as you can see, cohort is the only covariate with a significant p-value, and it has the largest hazard ratio. So since batch effects seem to potentially be driving our model, we decided that for the next steps of our analysis, we would analyze the cohorts separately. So the next thing that we wanted to explore was whether or not specific copy number alterations were occurring repeatedly across samples. And if so, were any of those alterations correlated with changes in survival? So to do this, we ran GISTIC, which is a tool that takes in copy number calling results from a set of samples and identifies regions that are significantly recurrently altered across the samples. So for each of these recurrently altered regions, GISTIC reports a Q value for the false discovery rate. So that allows you to choose the cutoff for your significance threshold. And it also reports the start and end positions for the genomic location of each recurrently altered region and provides a list of genes contained within that region. So we ran this tool on each of our cohorts separately. And because of the small sample size of the WashU cohort, we decided to use a fairly lenient Q value cutoff to allow us to detect more potential events. So this heat map is showing the copy number values for each significantly recurrently altered region that was detected in the WashU cohort. So each row in this heat map corresponds to one of the samples from the WashU cohort, and the samples are grouped by their diagnosis. Each column corresponds to an amplification or deletion. So the columns with maroon bars at the bottom are regions that are reported as recurrently amplified, and the regions with the light blue bars are regions that are reported as recurrently deleted. And each amplification and deletion is named by its chromosomal location. So in the heat map, the red spaces correspond to copy gains with a darker color indicating greater copy number. And then the purple spaces are copy losses, again with darker color indicating greater loss. And the white spaces indicate that a particular alteration was not detected in the sample corresponding to that row. So in total, in the WashU cohort, GISTIC detected 24 recurrently altered regions at an FDR cutoff of 0.25. So this heat map is showing the copy number values for each recurrently altered region that was detected in the CBTN cohort. So similar to the last plot, rows correspond to samples, columns correspond to recurrently altered regions. And because we have so many more altered regions in this cohort, unfortunately, we weren't able to include those labels of chromosome position. But in the CBTN cohort, in total, we identified 234 regions that were significant at an FDR of 0.25, and 220 of them remain significant at a cutoff of 0.5. Or sorry, 0.05. So after identifying these significantly recurrently altered regions in each of our two cohorts, we next wanted to determine if the copy number changes in any of these regions were associated with changes in survival. So to do this, we used the Cox proportional hazards model again. So for each recurrently altered region, we ran the model with the copy number values of that region as a covariate. So in the WashU cohort, we identified two regions of deletion that were significantly correlated with changes in survival. So those are shown in the table at the top of the slide. So one on chromosome 11 was associated with worse survival, and one on chromosome 15 was correlated with better survival. And then in the CBTN cohort, we identified 17 recurrently altered regions, 8 amplifications, and 9 deletions. So similar to the WashU cohort, we saw that some of these regions appear to be associated with worse survival, while some were associated with better survival. And then the next thing we wanted to look at was if there was anything interesting going on in these recurrently altered regions. So the next thing we decided to look at was whether any of the regions that were associated with changes in survival contain any previously reported cancer genes. So as I mentioned previously, the output that GISTIC reports from each region includes a list of genes that are located in that region. So to look for known cancer genes in these regions, we simply cross-referenced the list of known cancer genes reported by Cancer Gene Census with the list of genes reported in each region by GISTIC. So in total, we found 4 recurrently altered regions in the CBTN cohort that were associated with changes in survival and contain known cancer genes. So that's what's shown in this table here. So this plot is showing a clustered heat map of the 4 recurrently altered regions in the CBTN cohort that contain those known cancer genes. So again, rows correspond to samples, columns correspond to the altered regions, and the list of the known cancer genes found in each region is listed underneath. So in this heat map, the rows and columns have been clustered based on the similarity of their patterns of copy gains and losses in these 4 regions. So as you can see, many of the samples don't have gains or losses in any of these regions, but those that do seem to show some patterns. So for example, many of the samples that have a loss in REC-QL4 don't show any evidence of gains or losses in the other regions. And additionally, there doesn't appear to be any clusters that are primarily driven by diagnosis group, suggesting that whatever patterns are found within these regions occur across the diagnosis groups. Obviously, though, additional analysis will be needed to explore whether or not any of these patterns in these 4 regions are associated with any changes in survival. So just to recap some of the main conclusions from our analysis, we found that copy number alterations were more common than expected, and in several cases quite extensive. And although we did not see evidence of correlation between the extent of copy alteration and survival, there were some recurrently altered regions where we identified possibly some correlation with survival, including those 4 regions that contained the known cancer genes. Although, of course, additional analysis will be needed to look at that. And then a couple of the future directions for this project are to explore those recurrently altered regions further. And we're also working to harmonize the copy number calling for our 2 cohorts. So as I mentioned, the WashU cohort had copy number calling performed with CMV kit, while the CBTN cohort had calling done with control-free C. So currently we're running CMV kit on the CBTN samples, and again, performing manual curation of those copy number calls. And then we also plan to run control-free C on the WashU samples following CBTN's method. So hopefully the results for our 2 data sets will be more similar and reduce some of that batch effects that we see. And then finally, I'd just like to acknowledge our lab and especially our collaborators, Josh Rubin and Chris Miller, who provided the samples and initial analysis for this project.
Thank you, Sharon. So I'll just start with the questions. The first one is, how did you factor in tumor cellularity? Do you have a pathologist who sort of screened it for tumors that had very high percentage of tumor cells, or were they like 100% tumor? Because I'm guessing that it would impact how well you can call CNVs.
Yeah, definitely. Unfortunately, we don't have any pathology results. We have discussed using some computational tools, though, to look at tumor purity, because we are seeing some samples where there's definitely, I guess I'll say interesting patterns in the copy number calling that we're a little concerned about.
Okay. Yeah, because I'm just wondering, like, in terms of being able to apply this, like how much tumor cellularity will, determine copy number calling capabilities if you have a specimen that has a very low tumor cellularity, whether that would impact.
Oh yeah, that would definitely cause an issue. There are some tools where you can include an estimate of tumor parity in the parameters, so it can take that into consideration.
Yeah.
So yeah, that's a great point. That's definitely something we're considering.
Okay. Next question is, were there any novel copy number findings that didn't go along with the previously expected copy number changes for the tumor subtype?
Let me think. There was not technically a copy number, I guess, but some evidence of a structural variant that we saw across diagnosis groups on chromosome 17. So maybe not necessarily unexpected given it's chromosome 17, but definitely worth exploring more. So we're actually looking at including the structural variant results right now.
Okay. So another question is, these CNVs, are these CNVs de novo or inherited from the parent? I'm just sure this is just one of the questions I'm seeing here.
These CNVs are generally somatic, meaning they're only found in the tumor, most likely. Obviously, we can't say that with 100% confidence since we don't have matched normals for the WashU samples, but generally what we're trying to call is things that would be unique to the tumor.
Was CNV percentage associated with any other genomic feature, such as sequence variants?
So we haven't looked into that yet, but that is something we plan to do. So great question.
Okay. And was the CNV percentage associated with tumor cellularity? So again, related to the very first question.
Yeah.
Okay. And then one other question is, how different are copy number changes in pediatric cases versus adult brain tumors?
So they're definitely more common in adult brain tumors just because obviously adult brain tumors, you've had a lot more time to acquire mutations. And so you seeing less genomic stability isn't unexpected if you're an adult, I would say.
Okay. I think the final question here, for the medulloblastoma, there are at least 4 different subtypes. Each has a different copy number profile. So is there a thought to subgroup the medulloblastomas?
Yes, yeah, that's a great point. We have talked about subtyping. I think we'll need to increase our numbers a bit to be able to do that with any sort of power. But yeah, that's definitely another point that's been brought up. So good question.
Okay. All right, I think that's about it for, I just see some comments, but not really questions. I think that's about it for questions. Thank you very much, Sharon and Nicholas, for discussing your work. And if we do get any other additional questions, we'll just direct it in your way. But thank you very much.